So here's talk 2 that I gave at the Hospitalist and Resuscitationist Conference a few days ago in Montreal. And the title is Back to the Acid Basics. Is lactic acidosis a myth? So again, it's going to be the same case as the first talk. It's a 63-year-old woman with some social cigarette smoking who came in with the community-acquired pneumonia that we saw in the chest CT and self-treatment with chicken soup, which led to some subacute pulmonary edema. She's obese. She came in wheezing with lower extremity edema. And as you recall from the first talk, her BNP was mildly elevated, but so too was her lactate. And she had some atrial fibrillation. She received the cocktail in the ED, and she had the CT angiogram, which we saw that showed the lingular infiltrate, bilateral pleural effusions, geographic mosaicism, and central lobular nodules with interlobular septal thickening. So you saw her the next morning, and she was still hypoxemic and working hard to breathe. The question is now, with this information of the elevated lactate, what should you do? And let's assume that you started her on some high flow or some CPAP based on the first lecture. So now, obviously, because of the elevated lactate, you should give her some more fluids to help tissue perfusion from the pneumonia and sepsis that she has. And so I'd like to make a case why this is obviously not the right answer. So the objectives for this talk is to appreciate the difference between anaerobic and hypoxic, to understand the formation of lactate and its relation to acidosis, and to humor me briefly regarding the mechanism of lactate elevation. Also, to appreciate the many reasons why lactate falls with fluid administration, and that lactate may actually be beneficial. So I'm going to start off with a bit of a pet peeve and a bit of a rant, but it's this idea of aerobic versus anaerobic glycolysis. So you're going to encounter many resources that say that glycolysis can be both anaerobic and aerobic, depending on the presence of oxygen or not. And that's just simply not true. Glycolysis is always anaerobic. Glycolysis was going on for many, many, many millions of years on planet Earth before there is any such thing as oxygen as a terminal electron acceptor. Every single enzyme, from the conversion of glucose to pyruvate, does not require oxygen. Glycolysis is always anaerobic. So there's this tendency, as I mentioned, to call glycolysis aerobic when it's in the midst of oxygen, but this is highly anthropocentric. Glycolysis is an ancient evolutionary pathway. So as an example, think about your red blood cells, right? So red blood cells are clearly immersed in oxygen. They're essentially just a sack of hemoglobin saturated with oxygen, but they also require some ATP, and the only way that they can do that is through glycolysis. But there's no mitochondria, there's no terminal electron acceptor oxygen, there's no Krebs cycle. Is this aerobic glycolysis because it's in the presence of oxygen? No, it's anaerobic glycolysis. It's always anaerobic. So let's get on to lactate. So the question that I'm going to pose is, does the formation of lactate elaborate protons? Because if we go with the Bronsted-Lowry idea of acidosis, is that in order to create an acid, you really need to elaborate protons and lower the pH. So if we think about that equation that we learned about many, many, many years ago in undergrad biochemistry, you start with a molecule of glucose, you then have some reducing molecules, NAD, you have ADP and some inorganic phosphate, and you go through the steps of glycolysis, and you can see with that exploding proton there, you get 2 protons that are elaborated, right? From the 6-carbon glucose, you get 2 3-carbon pyruvates, you generate a net of 2 ATP, 2 NADH, and 2 protons. So that makes sense, right? You get pyruvic acid out of glycolysis. But then, those 2 protons are consumed, right? So you have a net 2 protons produced during glycolysis, and then you have a net 2 protons consumed to make lactate. And teleologically, that kind of makes sense, right? In the setting of aerobic respiration, true aerobic respiration, when you have oxygen as your terminal electron acceptor, when it's a glycolysis only, or in the absence of oxygen, ends up actually being pyruvate. So it ends up consuming the electrons and the protons and getting shunted into lactate. So there's an even proton balance here, right? So then stoichiometrically, how do we get a lactic acidosis? And so let's talk a little bit about how we normally classify lactate elevation. So there's also a C-type, which I'm not going to get into because I think it's just silly, but we'll talk with what we're commonly kind of force-fed in medical school. There's two types of lactate, and A-type lactate is what we're thinking about in terms of sepsis, right? There's starved tissues, the Krebs cycle is dead, there's hypoxemia, there's tissue dysoxia, and there's no mitochondrial respiration. And so the lactate goes up in this situation. And that's what we're treating when we see a patient with pneumonia and lactate. So the reflex, if you think of it, and if you think of lactate only in this terms, the reflex is that you must give fluids because you must augment cardiac output and therefore oxygen delivery to feed these starved tissues. And then the B-type lactate is essentially lactate elevation in the setting of anything else. And there's a list as long as I am tall of different reasons why lactate is elevated. But what I'd like to point out is that B-type lactate is probably the most common kind of lactate elevation that you're going to encounter on the floor. And I'd also like to make the point that these two are not mutually exclusive. You're completely allowed to have both A and B going on at the same time. So let's talk a little bit about glycolysis and Krebs, right? Going way back to biochemistry. So it's this idea of A-type lactate, which we tend to think as the major culprit in sepsis, where you start with glucose here, you go through all the steps of glycolysis that I just talked about, and you get 2,3-carbon pyruvic acid molecules because you elaborate these two protons, and then pyruvate goes through pyruvate dehydrogenase, and it gets turned into acetyl-CoA. In the Krebs cycle, it spins around, you pull off reducing equivalents, NADH, NADPH, and the protons and the electrons are fed into oxidative phosphorylation, and oxygen acts as the terminal electron acceptor. It gets turned into water, and protons are pumped into the inner mitochondrial membrane to drive ATP synthesis. So we all remember this, right? So in A-type lactate, what happens? Well, the tissues are starved of oxygen, so oxidative phosphorylation goes away. This backs up and destroys Krebs cycle. The Krebs cycle stops spinning, right? And then pyruvate is shunted to lactate, and the lactate to pyruvate ratio goes up. Normally the ratio, I believe, is about 10:1. But also, as I talked about stoichiometrically, this then consumes those two protons, and so then the question becomes, well, then how do we get the protons in type A lactate? And what probably is happening is that all those protons that were being jammed into the inner mitochondrial membrane as a consequence of oxidative phosphorylation then leak out and needs to be buffered by bicarbonate in the tissues. And when the protons are buffered by bicarbonate, the venous effluent, venous carbon dioxide levels go up in terms of millimeters of mercury. So this is your classic A-type lactate. Well, what about B-type lactate? So let's start with the same biochemistry. In this situation, we have glucose, but let's say there's simply high glycolytic flux, and glycolysis is normally much, much faster than the Krebs cycle. So say there's some sort of stress where glucose is degraded to pyruvate at a very rapid rate. The Krebs cycle and oxidative phosphorylation are still functioning normally, they're just much slower. So in this situation, again, it is quote-unquote a proton neutral process, but lactate starts to rise in the setting of just high glycolytic flux. So the ratio between the two tends to stay the same. And you can see chemically, you have a carboxylic acid group here in pyruvate, right? A carboxyl group here. And then if you don't believe me, here are the two protons coming out from glycolysis, and then the two protons actually then get consumed and turned into this alcohol group here as the pyruvate is reduced by lactate dehydrogenase. So again, Krebs cycle fully functioning, lactate's rising, it's just because it's really fast. So then the question is, well, where do the protons come from in that last paradigm in the B-type lactate elevation? Well, there's debate about this, and if you're interested in the nuts and bolts of the debate. You can check out the Great Lactate Debates Part 1 and 2 on pulmccm.org. But I'm going to go with the definition that's endorsed by the recent 2014 review in the New England Journal of Medicine on lactic acidosis, which is that work is what causes the proton elaboration. So if you remember, if you start off with ATP and then you do some biochemical work, you then hydrolyze that ATP into ADP, inorganic phosphate, and a proton. And then if you do even more work on the ADP, you get to monophosphate, inorganic phosphate, and the proton. So how might that look in the cell? Well, here's a very fancy slide that took me many hours to make, and we're going to start over here with the adrenergic receptor. So stress, any sort of stress on the body results in adrenaline and noradrenaline release. And what that does is it stimulates the adrenergic receptor, which then acts through an activated G protein to stimulate adenylate cyclase here. And adenylate cyclase consumes ATP, so this is biochemical work, right? The ATP is then turned into cyclic AMP, and the cyclic AMP acts as a second messenger to stimulate the sodium-potassium ATPase. And in the name sodium-potassium ATPase, we know that it is hydrolyzing ATP into ADP, inorganic phosphate, and a proton. And similarly, the ATP that's consumed by adenylate cyclase is turned into pyrophosphate, which is then consumed by pyrophosphatase into 2-organophosphate and a proton. And for those of you who are interested, it is this enzyme that is inhibited by caffeine. So this is part of the reason why caffeine makes you feel the way it does. And the cyclic AMP is hydrolyzed into AMP and proton. So this is the biochemical work that's occurring. And this pathway is really needed from kind of an evolutionary perspective to keep the tissues ready to fight or flee. So all of these second messengers and pathways keep muscle cells ready to act. And so this leads to the increase in heart rate, the contractility, and vasomotor tone. So this is an important pathway to have running in response to stress and epinephrine. And there may actually be within the cell different subpopulations of glycolytic enzymes that are specifically responsible solely for keeping these cell membrane activities rolling. But as you can see, as a byproduct of this stress response, you have ADP being generated and ATP being consumed, and you need these to be regenerated to keep this whole process moving forward. So how does that occur? Well, that's glycolysis. So remember, ADP is a substrate for glycolysis. You have to have ADP for glycolysis to move forward, and the ADP can come from these cell membrane processes. And so then the glucose and the ADP are turned into the lactate, which the pyruvate, which then consumes protons into becoming lactate, and then you get 2 ATP, which that can feed back and keep this process going without any need of oxygen. Even though in this particular cell, the Krebs cycle could be rumbling along perfectly well. This is your anaerobic glycolysis and lactate formation. So this could be where the protons are coming from in a B-type lactate. But in this paradigm, the important thing is we would call this a biochemical work-induced acidosis with associated lactate elevation. And by the above, lactate-generated muscle is utilized by the brain and heart as a source of metabolic carbon. And there's good data to show that during times of stress, the brain actually, kind of the way it does with ketone bodies, turns to lactate as a source of energy. There's also fascinating data that shows that the heart turns to lactate during times of stress, and lactate on the heart can actually increase contractility, and lactate can increase cardiac output. So lactate is probably actually an adaptive stress response and a very important molecule in terms of carbon shuttling around the body during times of stress. And also, what should be clear is that lactate is a marker of metabolic stress. It doesn't necessarily require that tissues are starved of oxygen. And so in this paradigm, the rise in lactate simply mirrors the increase in adrenergic stress. So the differential diagnosis of a lactate elevation should be really no difference no different from the differential diagnosis of an elevated heart rate, like sinus tachycardia or fever or any of the other things that accompany adrenergic stress. So, for what reasons might our patient's lactate be elevated without invoking any tissue hypoxia or that so-called A-type physiology? Well, our patient was working extremely hard to breathe because she was drowning, and hypoxemia is a very, very potent stimulus of adrenergic stress. So, she may have had a lactate elevation simply from hypoxemia. The muscles of respiration were working very hard, which were consuming oxygen and which were stimulating or at least following those biochemical processes outlined a few slides ago. So if you think about it, simply giving her high flow or BiPAP or CPAP and lowering the work of breathing and taking the stress off of her muscles of respiration in and of itself could lower her lactate because it would be lowering the metabolic stress. You can imagine, and I'm sure you've seen these patients get positive, non-invasive positive pressure, and you see their heart rate drop from 130 to 80. Well, you'd also, because of the release of adrenergic stress, you would expect the lactate to fall. Additionally, the patient received a lot of inhaled beta-adrenergics, which by that previous pathway is a sympathomimetic and can raise lactate, which is a common boards question. So you can also quite reasonably, based on these mechanisms, realize how giving her more fluids reflexively for an elevated lactate could make her lactate worse. And that would be because her lungs are underwater, because she's drowning, her lactate is up, and if you reflexively give her fluids, more of that will back up into her lungs. She will work even harder to breathe, and her lactate will get worse. You're not feeding any starved tissues, you're just making her work of breathing worse. So is there a way to distinguish between A and B type lactate? Well, not really, but this is kind of a stylized and theoretical way to think about it. Again, I can't emphasize enough how this curve is highly varied. It's a bit like the Starling curve. It will be different between different disease states and different patients. So none of these values should really be taken at face as if they're sacrosanct. This is purely for illustrative purposes. So this is oxygen delivery on the x-axis, oxygen delivery falling in this direction, and this is oxygen consumption on the y-axis. And so you've probably seen this curve before. And as cardiac output, let's say, starts to fall and oxygen delivery starts to fall, you could very easily have a lactate elevation, say, in our patient in this case, because of her stress response. And in this situation, I would call this type of lactate a B-type lactate elevation. Oxygen delivery is falling, but oxygen consumption is staying constant. And when that's occurring, what you know was happening is that the tissues are extracting more oxygen. They're extracting more oxygen to keep the VO2 or oxygen consumption constant. So if you had a VBG downstream of where you've drawn that VBG, you can make some biochemical inferences. So as delivery starts to fall, oxygen extraction ratio goes up. So the saturation in percent on the venous blood gas would fall from a normal, say, 75%, because there's normally a 25% oxygen extraction ratio, and it just continues to fall. But what happens is, as mentioned in the previous slides, is that as oxygen is consumed, carbon dioxide is produced. But it does so because its oxygen VO2 consumption is constant. Carbon dioxide production is also constant. So they fall, but they fall on the same ratio. And the ratio is the respiratory quotient, which we're normally taught is about 0.7 or so, but it's less than 1 in this area. You will get to a point, however, wherever this is, it's usually a very low number in both healthy and sick patients, but you're going to get to a point where tissues truly are starved of oxygen. So let's say you've thrown a clot, an arterial clot, to your arm, and all the tissues downstream of that clot are truly hypoxemic, and the Krebs cycle has stopped functioning. So in that situation, what then starts to happen is that the tissues consume less oxygen, so the saturation actually starts to rise, right? The saturation will rise because the oxygen extraction ratio is falling, because those tissues are just not using the oxygen. So then you can imagine that the VO2, our oxygen consumption here on the y-axis is falling. It's truly falling, as opposed to here where it's constant. So what this does is it starts to raise this quotient, right? And similarly, as shown in that type A slide, the venous carbon dioxide continues to rise as those tissues are starved for oxygen, because those protons that are, say, leaking out of the inner mitochondrial membrane need to be buffered by bicarbonate. And so what that does is it leads to an increase in the PvCO2. And so you get this increase in carbon dioxide, I'll call it production, but you have an increase in the carbon dioxide effluent in the tissues and a decrease in the oxygen consumption. And so the ratio starts to go up much greater than 1. And there's been a couple of neat physiologic studies in the last couple of years that are in a post that are linked to in a post that I entitled Rising Lactate and the Art of Venous Blood Gas Interpretation, and you can find those there. So again, the objectives of this talk is to appreciate the difference between anaerobic and hypoxic, to understand the formation of lactate and its relation to acidosis, and to humor me a little bit as I dive into the nuts and bolts of this chemistry. There are many reasons why lactate falls with fluid administration, and it doesn't by any stretch mean that you are augmenting oxygen delivery to starved tissues. It could just mean that you're mitigating the stress response. So please check out these other references, and thanks.